Think about what is happening inside your skull right now. Not your thoughts. Your brain's chemistry. Its age. The slow drift, neuron by neuron, toward a version of you that forgets more, processes less, and connects more slowly. Most people assume that drift is inevitable, a fixed biological clock. But what if part of it was simply a response to how often you eat? Scientists at the National Institute on Aging wanted an answer, not a theory, a measurement. So Dr. Dimitrios Kapogiannis and his team recruited 40 older adults, split them into two groups, and gave them 8 weeks. One group followed a 5-2 intermittent fasting protocol. 2 non-consecutive days per week, they ate just 480 calories. The other 5 days were unrestricted. The second group followed a healthy Mediterranean-style diet with no fasting at all. Then the researchers scanned their brains. The results were published in Cell Metabolism in June 2024, and they were not what most people expect. The first finding was about age. Not your age, your brain's age. Every brain has 2 numbers, the age on your birth certificate, and the biological age that shows up on an MRI, how old your brain structure actually looks. Scientists call the gap between them brain age. A high brain age means your brain looks older than it should. That gap is linked to cognitive decline. It predicts it. After 8 weeks, the brain age scores dropped in both groups. The biggest changes appeared in 2 specific regions, the anterior cingulate cortex and the ventromedial prefrontal cortex. These are not peripheral areas. They are command centers. The regions most responsible for decision-making, emotional regulation, and executive control. The fasting group showed numerically greater improvements here than the healthy diet group. The second finding was about glucose. Chronically elevated brain glucose is one of the quieter problems of modern eating. When your brain is flooded with more glucose than it can use, neurons function poorly. Cognitive decline accelerates. Using magnetic resonance spectroscopy, Kapogiannis and his team measured brain glucose directly. After 8 weeks, concentrations dropped in both groups. The brain was becoming more efficient, burning fuel cleanly rather than swimming in excess. The third finding is the one that matters most for long-term brain health. Insulin resistance in your neurons. Your brain cells need insulin to work, to absorb glucose, to communicate, to clear out metabolic waste. When neurons become resistant to insulin, that system starts failing. It is a recognized risk factor for Alzheimer's disease, and until recently, measuring it in a living human brain required invasive procedures almost no one would volunteer for. The team at the NIH found a way around that. They measured tiny particles called neuron-derived extracellular vesicles, microscopic fragments released by brain cells that circulate in the bloodstream. By analyzing these particles, they could assess insulin signaling inside your neurons without ever touching your brain. In both groups, neuronal insulin resistance dropped significantly. The fasting group dropped further. Your brain was getting better at listening to its own signals. Executive function improved as well. The ability to plan, to hold multiple ideas at once, to solve problems without losing the thread. The fasting group outperformed the diet-only group on this measure. Blood levels of beta-hydroxybutyrate and acetoacetate, the 2 main ketone bodies, rose higher in the fasting group. Consistent with the deeper metabolic shift that comes from eating only 480 calories on fast days. Now, here is what is worth sitting with. This was not aggressive fasting. It was 2 days per week of serious calorie restriction. The other 5 days were completely normal. No special foods, no macros to track, just 2 days of eating very little. And in 8 weeks, the brains in this study looked measurably younger. Neurons were more sensitive to insulin. Planning and problem-solving improved. The biological machinery of the aging brain had shifted. This does not mean fasting prevents Alzheimer's. Kapogiannis was explicit that this was a pilot study. The methodology itself was the point. A framework for measuring what diet actually does to the brain. Amyloid and tau markers in cerebrospinal fluid, the proteins most directly linked to Alzheimer's pathology, did not move significantly in 8 weeks. Longer studies are still needed. But something is clearly happening. Something measurable. Something biological. If you have ever noticed your thinking sharpens when you fast, that your focus feels cleaner, your planning more precise, this study gives you a reason. Not a theory. A measurement. The anterior cingulate cortex. Neuron-derived vesicles. Brain glucose falling. Insulin resistance retreating. Your brain does not need to be fed constantly. Some of its best work happens in the quiet spaces between meals. That is what Dr. Kapogiannis and his team went looking for in those 40 people over 8 weeks. They were not measuring hunger. They were measuring your brain. And what they found changes how we should think about what skipping a meal actually does to you. If you want to go deeper, everything on fastinginpractice.com is free. Hundreds of articles built directly from published research, a global community of fasters, recipes, and tools to help you find your protocol. Visit fastinginpractice.com. The link is in the description.